I remember correctly, last 20 years during the JNU time. So it's really great. All the time I see that the way that you are nurturing the education system in India, nurturing the new generation by your knowledge of several subjects. So first of all, I like to really give thanks to you that it's really a very innovative job that you are doing for our academics in India.
अभी भी मैं सर गणपत यूनिवर्सिटी आया हूँ, जस्ट 5:30 लेक्चर खत्म करके आया उसी गेस्ट हाउस में, मेहसाना गुजरात में हूँ, गणपत यूनिवर्सिटी गेस्ट हाउस में हूँ.
Yes. Yes, so thank you very much. I can see that even during your study at university, you are a very active researcher. And I do respect all the mentor, especially Professor Aparna Dixit, Madam, remember today. So, great. So, I like to thank Poonam Madam for a very nice introduction about me to the audience. It's happened thank you very much. So,
Poonam Madam is assistant professor at Kiran Patel University, Baroda, and Global University. She is assistant professor and she is also PhD from IMTech.
Oh,
Chandigarh.
Great. Yes. So great, Madam.
Always help me. We all are in different cities, but always she is associated with me, and we all are trying to improve overall science.
Yeah, so it's great. Nice meeting you all here online. Okay. Yeah, because now this is a new platform. I hope that my slide is visible.
Yes, sir. Yes, sir. It's visible.
Yeah, so because when I was discussing with Dr. Gopal, then I thought that why not I should give a talk to like a faculty members and esteemed members who are attending this session, and especially for the students. So decided a topic nanotechnology for infectious disease. So it's a kind of combination of immunology and physics, chemistry, and of course the translational science. So the reason that I selected this subject because all the time this nanotechnology, all opportunity, many publication, patent, up and down. There are many things that we can debate, especially on the topic of nanotechnology. Infectious disease is a disease that because everyone is a scientist, this is our duty to work for the human welfare. And one of the problem that global problem that is the infectious disease problem. And this is a very simple definition that I want to start with students, that you know that when we are talking about nanotechnology, then nanotechnology means something related to nanoparticles. And nanoparticles, according to nanotechnology initiative, anything having a structure less than 100 nanometer in size is called nanoparticle. Okay, this is in like for metal nanoparticles, not for biopolymeric nanoparticles. And this cartoon generally it shows that if you have a nanoparticles like this white background, like 100 nano, even 10,000 to 20,000 nanometer in diameter. So it's very common, like even if we can say that not scientific way, then nano device, because being a very small device structure, it can easily enter inside the cell because it has 10,000 to 20,000 nanometer. And after entering the nano device inside the cell, this nano device can interact starting from cell membrane to cytoplasm to the protein to the nucleic acid, and then there are many things that we can see. But one thing is also very clear that now one thing is clear that size below 100 nanometer basically for metallic nanoparticle is called a nanoparticle. Second thing, this is also very interesting slide that I want to share, that when this concept of nanotechnology was started, being a researcher, and you know that we love the nature, and in the nature when we see the butterfly, a beautiful color of butterfly, different color of butterfly, and butterfly fly here and there. But when the scientists saw a butterfly, then realized that the beautiful color of the wings of a butterfly, it is a layer of nanoparticles, and that's why that's a kind of imagination that when a butterfly, it's not like a very strong or something like a, and it is surviving in the nature, in the rainy season or in the winter season or in the summer, and all the time it's due to the color. It makes the happiness to surrounding. Now, this was a concept when people thought, okay, the layer of the wings is, because more than that, also go to the second slide. If see like a butterfly wings, then it's inspired the scientists, and that was a kind of start of assembling of textile and applying the approach. But again, come to our mind that okay, if this is a matter of a size of 100 nanometer, then people thought that there are two approach of having the to making the nanoparticles. Generally, one is starting from atoms, iron, and molecules, like they can combine together and make the nanoparticles. There is another way that we can have a bulk precursor, and we can like make a small and make the nanoparticles. So it's a top down, top up, and bottom up approach. There are two approach, theoretically or in the lab, that we can think that this can be made. But altogether, one thing that I want to make clear here is that because this nanotechnology is a technology that is more oriented towards the scientist of the physics background and the chemistry background, as a biological people who are doing the research, because now this is an era where we are talking about interdisciplinary research, interdisciplinary knowledge. There is nothing we can say that I am a biologist or physics or chemistry separated. So we have a platform that where we need to share our idea. So that is good that okay, slowly when the biologists realize that okay, this is a approach that physics can defining the nanoparticles, and chemistry are applying that this is a top and bottom up approach of making the nanoparticles. Then we applying in the biological sciences. So generally, in the just for a student, I want to explain that when we talk, when we take a nanoparticles in the drug delivery. So this is a cartoon I have taken somewhere from paper, that there are generally four steps when nanoparticles enter inside the cell. The first step that we need to incorporate the drug or encapsulate the drug inside the nanoparticles. After having the nanoparticles into the drugs and nanoparticles together, they bind on the surface of the cells, and through a process endocytosis or phagocytosis, this drug enter inside the cell, and after that we know that inside the cell we have like a lysosome, and there is a fusion of phagolysosome or endolysosome, and this endosome or the phagosome gets dissolved inside the cytoplasm, and whatever the content that that we have encapsulated before making this like a drug delivery system, it has their own way. So if you can give and see the steps here, then one is the preparation of nanoparticle carrier. Second is basically the binding agent go and recognize the target cell. The third step is go inside the cell, interact with the biomolecule inside the cell, and the fourth is its breakdown, and then whatever the pharmaceutical agent, for example, it release and execute the job. So this is a picture I have taken from UPES State University of State, and that was very interesting paper I read in 2007 that was published in Nature Nanotechnology. And that was really fascinating for me to see that when nanoparticles are made in the lab, because we have something that we can control the size, we can control the charge, we can control the hydrophobicity, or we can make them targeting. So it was reported in that paper that when the size is more or size is less, then there is a different Th1 and Th2 stimulation, like for immune system. It has a different adjuvant property like hapten and all this. So the take-home message, what I want to discuss here is that these nanoparticles, which is made or applying the nanotechnology in the lab, it is not only for drug delivery or other application, because when we are going to see the application part, there are many applications, but it depends on our area of research where we can apply these nanoparticles. So generally, as an immunologist, when we see it, then many times it's become very important when we think about targeting for treatment of certain diseases. And at the same time, we also have like a different type of immune system in the body. And if particularly if any immune system is getting upregulated or it is getting like a downregulated, then for an immunology point of view, it will be very helpful to set up an experiment in the lab, because now altogether this slide I want to make a conclusion saying that when we have a different size of the nanoparticles, then probably based on the size or the charge or the hydrophobicity, our immune system have a different way of acting. Now I come to a problem that we have done in our lab in 2013, 11 years back, that seen this paper, because I am not showing my other work. This is a work that is interesting for the student. That's why I thought that let's discuss this simple experiment, because now our concept is that because my title of today talk is nanotechnology and the infectious disease. Nanotechnology means nanoparticles having size less in the 100 nanometer. Then also we have seen that it has a very good way going inside the cell. When it is going inside the cell, then for immunology point of view, it is also showing that it has a different way of interacting with our immune cell. Now come to a problem, this is a problem of a health. Typhoid disease is a typhoid fever caused by Salmonella typhi, and this is caused by a bacterium Salmonella typhi. And we know that this is due to contaminated food or water, and this is coming from the sewage system or poor sanitation, eating food or drinking. Like these are the causes. This is like a common causes. Then based on this, what we set up a experiment in our lab. This is a very simple experiment, 10 plus 2 plus 3 a student like BSc level, that CFU, that colony forming unit. So what we have taken the THP1 cell, that is a macrophage cell, and cells were infected with Salmonella typhi. So it's a kind of in vitro model. And we have decided to check the colony forming unit at three different time points, like post 4 hour, post 24 hour, then after 48 hours. And then this data shows that if you see one was the control, another was very interesting that we have added the ATP, that is the energy currency in the human body. And then you can see here, when there is a control, then all the time point, the Salmonella colony were increasing. But at the same time, when ATP was added, the number of colony were higher in a group where ATP was supplemented in the media. So this was a kind of indication for us that okay, ATP has a certain way of controlling the growth of salmonella inside the cell. In the other way, we can say that okay, if ATP is there, then probably there is a role of mitochondria is there. Then probably all the metabolic pathway. So this a simple experiment tell us that okay, this is like a salmonella. Okay, in fact, the human body, we have set up a model in the lab and see that when the salmonella was growing, then at a different time point the number were higher, but ATP was supporting. By this message, we thought that okay, if the salmonella is growing very well, so I and our group we discussed that okay, let's have something more interesting and find out that what is the control of salmonella. Then, by chance, we decided the HIF1 alpha transcription factor. So, HIF1 alpha is a hypoxia inducing factor. And more than that, it's also interesting to know that anytime when a cells get infected with any bacteria, depends on the type of bacteria, but most of the time, this type of factor, one of the factors like HIF1 alpha, is become very important for study further and correlate with the metabolic pathway. So this data clearly shows that again this is very simple experiment, y-axis versus x-axis, and the cells like without infection and cells infected with salmonella type E, and you can clearly see that it was quite significant that cells were infected and CFU number was higher. Then at the same time, we also checked that when the cells were infected, when the colony were very high, then HIF1 alpha expression was also very high by doing the real-time PCR. So this simple experiment tell us that okay, when the salmonella is growing inside the cell, at the same time, HIF1 alpha is also inducing and its expression is higher. Then again, because I am going to correlate everything just for our students here, that how a simple problem, simple experiment, we need to correlate and get an answer. Because we need to ask a very simple question. So our question was to set up a model where we can see that how the salmonella grows inside the cell. Okay. Again, come to here. This experiment showed that okay, now you can see three state of experiment. One experiment is showing that when cells were infected with salmonella type E, then the colonies were very high. At the same time, when the cells were infected and treated with silica, the reason that we have taken the silica because silica has some relation with the mitochondria of the cell. So we have taken the silica, then we see that okay, the CFU number is lower. Again, we have taken another chemical agent like ecriflavin, that is also something related with HIF1 alpha or mitochondria or metabolic pathway, and it was also shown that the number of bacteria was less. So if you see three bar graph, then it shows that okay, when cells were infected, then the colony were very high, but a certain two like silica and ecriflavin both are reducing the number of colony. Then come to the next when we go for real-time PCR assay to check the gene expression of HIF1 alpha, then we saw that in the case of infection, the HIF1 alpha was, of course, higher. In silica was lower, but in ecriflavin it was almost negligible, like at the bottom line. That was for us was very interesting to see because I am just showing here the singlet of experiment. We have done like 4-5 times just to make sure that our experiment is working well. After we have also checked the IL-1 beta cytokine because this is simple ELISA. So IL-1 beta is a pro-inflammatory cytokine, And it also shows that, okay, when it has an infection inside the cell, that is IL-1 beta is higher, like a titer was very high. Then we also check the hexokinase. So you know that hexokinase is the first enzyme of glycolytic pathway, and it has outside mitochondria, but it's the first step that we can see it for metabolic pathway. And we check the hexokinase by real-time PCR and see that during the infection, the hexokinase level were very high. In silica was less, any acriflavine, no expression of hexokinase. So now, how this experiment is important for us? That one is a simple microbiology experiment, that is CFU. Another is like a real-time PCR. Another is ELISA. Another is the real-time PCR. And this inside, this is the flow cytometry using the facts. And then we check that, okay, when the cells are normal cell and the infected cell, and then probably we want to see some marker that with the mitosocs, like a mitochondrial superoxide. So as we know that in mitochondria, when we have a normal way of functioning and synthesis of ATP, then we need to have proper functioning of oxidative phosphorylation. But many times, if there is dysfunction in this pathway, then there is a release of mitochondrial superoxide. And that is a kind of indication that our mitochondria is not healthy or mitochondria channel has some problem. So we try to look for mitochondrial pathway where infection were there and to correlate whether salmonella has something to related in ATP synthesis or something like that. And what makes her more to look for that, you can see here that when cells were infected and when the acriflavine was there, then the mitosocs was quite higher. Okay, so based on that, again, this is a biological experiment. This is an infectious biology experiment in the lab, because everyone know typhoid disease is very common everywhere. And this is the nanotechnology. So we made the ketosane nanoparticles in the lab. So whoever has expertise here, they know that there are certain instruments which is which is required for measuring the size of the chitosan, for any nanoparticles. So we measure the size of nanoparticle using the dynamic light scattering, having 319 nanometer, because this is a biopolymeric nanoparticle. We have checked the size and correlate with transmission electron microscopy. So it showed that, okay, the size was 250 to 300 nanometer, and also measure the charge of the particle having 41 millivolt and plus minus 5.29. So this is of course, altogether is the nanotechnology, how we made the nanoparticles. And then probably now we have parallel two set of experiment. One set is infectious biology, understanding our basic biological knowledge and applying in the research, applying in the translational science. This is nanotechnology. We have learned something from the physics and chemistry, made some nanoparticles, and then we said, okay, let's have something that is encapsulate this thing, encapsulate the drug or compound inside these nanoparticles. So see this data, and this data tells us that, okay, when this is a colony forming unit assay, this is a real-time PCR data of HIFO and alpha. So these CFU assay tell us that when the cells were infected, then when we have the compound 1, then probably there were no infection. At the same time, no HIF1 alpha expression. So generally, this is when we move forward, then we have selected two different strains of salmonella. As we know that salmonella typhimurium causes the typhoid basically in the mouse, and salmonella typhi causes the typhoid in the human. So we have taken like a two-way going for the study in the animal model. So we have taken this Salmonella typhimurium as well, and also we have taken the Salmonella typhi. But if you see both are the different species, but the result, the pattern of the result was the same. Like either for CFUSA or HIF1 alpha, or here is the CFUSA or HIF1 alpha expression. So after that, we filed the patent in 2015, and this patent was got published in 2020, 4 years back. So this was a kind of a. Am I audible?
Yes, you are audible. Poonam madam, आपका कोई question है? You have raised hand.
हाँ sir, yes sir.
Yes, please ask.
Please.
Sir, I just wanted to ask that whatever the specificity is, it is at the genus level only.
Right.
So are these nanoparticles, they are also they can also work in the different it's a gram genus which are also gram negative, like E. coli or some other bacteria. Can these specificities also for them, or it is just for the Salmonella?
Yeah, I think for our case, we have checked in Salmonella, and this is a result I am showing for Salmonella. But I think it will vary from bacteria to bacteria.
Okay. So every time if we are looking for certain infection to cure them with these nanoparticle, we may have to design the nanoparticle specific to that particular infectious agent.
Right.
Not
Yes.
something like it is taking care of a large diverse population of or the diverse genus of any bacterial species.
Right. Yeah, because it's this all microorganisms have their own way of metabolic and physiology, so they have different way of interacting with anything that is coming from the outside.
Okay, sir. Thank you, sir.
So then my slide is visible, please.
Sir.
Yeah.
Yes.
Yeah, so then we have based on our nanotechnology and infectious disease correlation, because one disease that we have seen is the Salmonella typhoid, because this is a typhoid here. Then we have taken another disease. This is a Chagas disease. This is a disease of Latin America. So when I was in Latin America in 2015, after applying this patent, then thought that let's try this concept in this disease. So one thing is worrisome that in Latin America, this is a disease, and last 100 years, still people are dying, and many countries in Latin America having this disease, and there are no new drug discovery, no new vaccine, no treatment. So for a researcher, it's always a question that why these drugs and treatments are not getting a success. So generally, the mystery of this disease is due to Trypanosoma cruzi, and this is a protozoa. And this protozoa, initially when it infects a human being, then it has a stay in the acute phase and goes in the chronic phase, and during the chronic phase the patient dies due to cardiac arrest. So this disease started in the acute phase like a simple experiment, and the patient died due to the cardiac arrest. So altogether this disease become very complicated, and this was our lab in Latin America. So sorry, thanks to Professor George Khalil and Professor Idisho Kunaneto, because we have a lab here. So generally, there are two drugs which are available for treatment of Trypanosoma cruzi. One is the nifurtimox and another is benzenidazole. And last 100 years, these drugs are only effective in the acute phase, but not in the chronic phase. More than that, it has also severe side effect, including cutaneous reaction and GI tract disorders. So when I was in India, I proposed let's try colchicine. As a cell biology, we know that colchicine is toxic for microtubule and tubulin protein. But even though I saw some of the paper published in 1996 and 2002, so we decided with Professor Edishu that select the colchicine and is able to cure this protozoa and finally the disease. Second, chloroquine is very popular drug starting from malaria and during COVID-19. That how it's effective against initially malaria and protozoan disease. So we proposed that let's try to make a combination of the drug like colchicine and chloroquine and benzenidazole, three drugs together, and using of course, the nanoparticles. And nanoparticles, we thought that let's take the gelatin nanoparticle. So we know that everything that capsule that we take, the coverage of gelatin, we made the gelatin as a nanoparticles and make a combination of drug like colchicine, chloroquine, and benzenidazole together, encapsulated into gelatin nanoparticles. And this was a nanoparticle preparation. Again, the nanotechnology, we have used the Foster method of making this nanoparticle and check the size, and that was 150 nanometer. And now come to our biological experiment. So what we have done here, we have again taken the THP1 cell, like the same like Salmonella, and another cell like HEK293, that is human embryonic kidney cell line, and Trypanosoma cruzi infection model, and the cells were infected. Trypanosoma cruzi, this is the cells, 24 well plate, and having a drug 1, drug 2, and combination of drug, and measure the IC50. So IC50, the inhibitory concentration, which can kill 50% of the parasite. And this data, took like 6 months, but I am showing you some interesting data here that what we proposed from India using the colchicine and chloroquine together drug in combination. So if you see here that there were two concentrations based on the other experiment that we have done, and it shows that when we have the combination of the drug here in HEK293, there were no parasite release, zero. There were no parasite release. Even in THP1 cell, the pattern was the same. No parasite was coming. So it was, for us, was very interesting to see that, we have reached at a point where combination of drugs is doing something inside the cell and the parasites is not coming out. Then we calculated the IC50 value, and this data shows that when we have a benzenidazole alone, this is a drug for Chagas disease, the IC50 value was less than 0.78 micromolar. But when we come to a combination of drug, it was not able to determine because there were no parasite at all. So this was a finding of Heg cell. This was a finding of THP1 cell. The pattern was the same. Then we ask another question that, let's see inside the cell what is happening when the parasites stay inside the cell. So we have taken the THP1 cell and infected with Trypanosoma cruzi. And after 48-hour infection, using the Elexaflor and Debris staining, you can see the beautiful color of parasite. 100% cells were infected because we counted the parasite and cell using the fluorescence microscope. And afterward, when we have seen that when there are different way of treatment of drug, for example, when cells were there, 100% cells, when we have taken the chloroquine 5 micromolar, then 6% cells were infected, colchicine 44. But when there is a combination of a drug, benzenidazole and colchicine and chloroquine together, 4% cells were infected. So here, if we can compare infected cell and 4% cell, or a drug alone and the combination of drug, it shows that okay, combination of drug has significant way of reducing the parasite, not only the extracellular, but here the intracellular parasite as well. Then we thought that okay, let's see whether this drug is working on the parasite or not, or only on the cells. So we have taken the parasite and treated all this drug after 2 hours was and infected with the cell. And this was a picture that showed that okay, this is the parasite using the GIMSA, that okay, no effect on the parasite. This was a infection in benzenidazole, 25 micromolar. Okay, so you can see this is like a morphology of parasite and how the cells look. I will directly come to a combination of a drug, because that was our interest. That okay, when the cells were infected, and when we have shown that okay, this is a combination of drug which has reduced the number of infection in the cell, whether it has effect on the parasite or not. But this data shows that no, not at all, because even the parasite which was treated with all this combination of drugs, so there is no effect on the parasite. So then even we have used a high throughput screening using the operator content imaging system, using a very fast way of understanding and seeing that 48 hours of infection. And it was shown that okay, when we have the benzenidazole against this infection model, the effective concentration was 8.6 micromolar. Okay, and in like when we are taking the combination of drug using this high throughput screening method, then if I can compare here that effective concentration 8.6 and 0.8, then combination of drug is so that the efficacy of drug has increased like a tenfold. Then we have gone for a experiment that was more interesting for us, using the Syrian hamster, taking the 120 hamster divided in different groups. So this was a kind of 1 year experiment, seeing that whether this combination of drug or individual drug. So if we see here our plan of the experiment, then the plan of experiment say that this is the Syrian hamster. After infected of 6 months, we measure the echocardiogram, also the electrocardiography, just to check that whether any effect on the heart. Then 7 months post-infection, we have given 30-day continuous treatment of the drug. Then after 11 months, we again measure the echocardiogram and electrocardiography. After 12 months of completion of the experiment, we have a different measuring the blood. Then we measure a different fibronectin protein, histopathology, and also immunological markers. And these were the group of our animals in the when we were working, having a control group, having a group of animals which were infected only, where having the group of animal infected and treated with the drug which is available in the market with the higher dose. Then there is a group of animal infected and infected with some optimum dose. Then colchicine. Then there is a group of animal infected and treated with only chloroquine, and there was a group of animal which were infected and treated with a combination of a drug. So this is an experiment, and this is a take-home message for our students here, that what the graph I am showing you here, this looks like a one graph, like a percentage survival, that how the animals survive in 1 year time. But this experiment, you can imagine, to get this graph, generally we took 1 year. So one day, 365 days passed just to get this data. And this data shows that, okay, when the animals were given the combination of a drug, no single animal were died. So this was for us was very much interesting and exciting that, okay, what is happening with all these three drugs? Because when all these three drugs were given to the animal, and even after 1 year, we are not going to see the biological mechanism or some other mechanism, but at least as a biology point of view, we can see that animals are not dying. So it's itself a very interesting observation as a biologist that, okay, this shows that, okay, this drug has something that is not toxic and it is promoting the better health of the animal, because this was my observation in the animal house. So this shows that, okay, when animals were infected, some animals die. All the group animals were died except the group which were received the combination of a drug. This was a histopathology data showed that taking the heart of the animal, we measure the fibrosis. And this fibrosis, using a very well-trained, like a doctor who has expertise in histopathology observation, and we have given a score in the heart fibrosis, and this data shows that if you see this is the without infection, this is the only infection, and this is a suboptimum dosage, and this is a combination of a drug. So a combination of a drug, and if you compare with the infection, there was a significant reduction in the fibrosis in the heart. So it was itself a kind of a new discovery, and that led us that at that time in 2017-18, there was no a single paper in that our line. Still we are working in this line. So finally, I would like to acknowledge all my collaborators here to get all this data. Professor Edesho Kuna Neto, Professor George Khalil, Professor Bianca. Mariota is my PhD student, Professor Karina, Dr. Bera, Dr. Alexandri, Professor Tapan from India, Dr. Alexandri, Kelly, Camila, Alexandri, Eden, and thank you very much. Now I am open for the question here.
बहुत अच्छा. Any question from any participants? No question. Any common question outside this topic also you can ask question to him if it is related to nanoparticle, which he has not covered, but if you have a question in your mind, so you can ask. Yes, Manjisa, you have raised hand. Do you want to ask question?
Yes, sir. Good evening, sir. Really very nice talk. So I would like to know something regarding nanobots, because again, I think it's related to nanotechnology. So I would like to know about it. I've heard about it.
Can you repeat again, nano?
Nanobots, sir. Nanorobots, we say.
Yeah. I think it's because these nano robots and nano because it's coming in the surgery, it's coming in the medical science, and it's still So this but this is basically. If I was also reading some of the article related to this, I found that there is a scientist in State America, in Stanford University and another university. They have developed this technology. After that, there was a scientist from Banaras Hindu University. She contacted me saying that is it possible to set up this type of model in India. I tried to set up, but I think still it is in the very preliminary stage. The main problem is because it's not only the nanotechnology, it's a association of electronic and communication engineering as well as artificial intelligence. More than that, the problem that we are facing, how to make sure the aseptic condition.
Okay.
Yeah.
Any attendees have any other question? A student can also ask. Hello. Yes, Puna madam, आपका mic on है?
Sir, there is a participant, Gitanjali Dang. She has raised a question. Is she in the meeting?
Gitanshi
Dang.
You can unmute and ask. नहीं, इनका क्वेश्चन नहीं है. अच्छा हाँ, yes, season. So आपको दिख रहा है चैट में सर?
Yeah, I can see the question. Gitansi has asked a good question that silica and ecriplevin must be a part of the metabolic pathway, or they may also be involved in mitochondria of non-infectious tissue. Isn't there are any negative impact there? It's a good question from the student side, but again, my answer will be anything that we try in the lab for doing the experiment, we always use the references. Means if somebody has done in the lab and what are the concentration, what are the dose, if somebody has used, based on that only we try in the lab. So because many times we know that if there is a role of mitochondria in the infectious disease, then our approach was to look for certain thing that can induce the mitochondria, certain thing which can deactivate the mitochondria. So silica, we have seen in the publication that silica has some role in the mitochondrial activation. Ecriflavin has certain role in the, mitochondrial deactivation. We know that there are two types of disease. One is the contagious disease, another is non-contagious disease. So, for example, if any disease that is not related to infection, but our mitochondria don't discriminate between infection or not infection, because it all the time need to work and generate the ATP for survival of the human being. So, my answer will be yes. All the drug has a side effect and negative impact is there, but our only way that how we are going to minimize is by using the permutation combination and reducing the dose titration.
Gitansi, are you listening? Good. So now any other participants have any other question? Any other feedback? Professor Vipul Patel, sir.
Yes, sir.
Yes, sir. You have any question or any compliment?
Good evening, sir. First of all, thank you very much for nice session. Unfortunately, I was in between there and out of joining due to some others meeting also. I would like to express my gratitude towards the very wonderful session, sir. Just would like to on small kind of lung, that is lung cancer, small nucleus. So in that perspective, how would we can consider that nanoparticle and carrier drug delivery system will make a good success in biotherapeutic point?
Yeah, thank you very much.
Yes, Dr. Ramendra Pandey, sir. So Vipul Patel, sir, is Dean in Pharmacy College, Maharashtra, somewhere. Yes.
Thank you.
We are working right now with the chitosan modification, biomaterial development, and somehow after screening around 70 molecules, we got success in almost 16 molecules, which are showing very wonderful antimicrobial activity. So far with the skip base containing chitosan with the fluorine group. And certainly we are also working with some another derivative, dibenzyledine. So which showing a very potent this anti-cancer activity. But now the challenging question is that their molecules, because the quantity which we found to be effective is in milligram. So would like to make them into nanoparticle with a certain range, which can effectively penetrate into the body tissue part for a better therapeutic action, and to reduce the immunogenicity and this kind of things for anti-cancer property.
Yeah. So first of all, thank you very much, Vipul sir, for joining this.
Thanks. It's my pleasure.
For your nice compliment.
Yes.
So You have asked a very relevant question related to the cancer. So, according to my previous experience, you know that the cancer is a major health problem, not only in the India, it's a global problem. Second thing is that there are drugs which are getting resistance and not working for the cancer. Let's say cisplatin is a drug for breast cancer. Now it is getting resistance. So for a scientist, it becomes a very question mark what we should do, because when we go for a discovery of a new drug, it's a very expensive and a very long way. So, my answer will be anything that nanoparticles or a new drug or anti-cancer drug. When we are going to apply for a like a mice model or cellular model or a human model, I prefer just to go before pharmacokinetics and biodistribution study.
Yeah.
Because what happened after having a good pharmacokinetics and biodistribution study, our life become very easier to decide that what should be the optimum dose or concentration we can use. For example, I was working basically on the adenocarcinoma, and we have taken the Taxol as a drug. Okay. So my experience was that when there was a black mice, and we were giving the Taxol injection like intravenous based on the publication, and at the same time we have taken another nanoparticles, and that nanoparticle was encapsulated into the that Taxol. So my observation was that when injection was given to the mice using the nanoparticles and drug together, there was a clear-cut reduction of the tumor burden in the mice. So two things I will suggest. One, biodistribution and pharmacokinetics, and second, based on the literature, the way of giving the injection, like a subcutaneous, intravenous, subdermal, or intramuscular. So that will clearly show the difference in the result. Okay. Yeah, I think maybe I have.
Yeah. Of course, sir. That's pharmacokinetic and dynamics will give the clear-cut regarding the drug distribution among the tissue compartment and how the therapeutic way we are monitoring that things. Yeah.
Yeah. Yes.
Thank you so much, sir.
My
The
pleasure.
nice thing is that today thank you, Gopal sir, also, because you are here and work. So we are.
हेलो
Yes, sir.
मेरा डिस्कनेक्ट हो गया था शायद.
Yes. Thank you, sir. Just would like to share you for arranging such a wonderful and nice session.
Yes. So Jyoti, you can ask question, or if sir, if you can see her question in chat box.
Yes. Okay, so I can see a question after delivery of the nanoparticle with drug into target site. What is the final fate of this nanoparticle? So, okay. So generally, for example, I will just share my experience. There was a time I was working with the liver cancer. So liver cancer using the drug, and when we make the nanoparticles, so we keep in our mind that that nanoparticles will have some configuration that will be targeted, because our human physiology, human anatomy is very complicated. So this is the advantage of having a nanoparticle, because during the synthesis we can make sure that it can go to the target site, depends where we want to deliver the drug. I think maybe I have given the answer to your question.
Second,
Sir, after the delivery of drug into the target site, sir, it will get separated, or what the desired genetic material is coated on that nanoparticle. So that will get separated. That genetic material can be used by the cell machineries for further gene expression, for further. What will it do in the cell, both in human cells and plant cells? What is the final fate, sir? I want.
Yeah. So you are right, because you know that when these drugs nanoparticle is made up of protein, with a wide peptide pathway is or drug or not. For example, the protein pathway depends on the type of nanoparticles.
Yes, sir.
Okay.
Sir, and also I have another question. Sir, for the delivery of this nanoparticle, which method is used?
Yeah. Right. So, like for example, if you are talking applying for a, we you know that anytime we don't directly go for human study.
Yes.
We go for cells, then we go for animals. So many times, for example, when we go for animals for intravenous or intraperitoneal, just depends of a type of nanoparticles, and if you need to see if any paper is reported, then accordingly we need to select what route is the best, intraperitoneal or intravenous.
Okay, sir. Thank you, sir.
Welcome.
Shruti, Shruti, are you here? Shruti, is it possible to block viral replication through nanoparticle? She's asking.
Yeah, so it's a viral replication and nanoparticle. So this concept of nanoparticle was started from retrovirus, because initially people were using the retrovirus for delivering of nucleic acid and drug inside the human body. But problem was that retrovirus, when it was going inside the body, when the time comes, it will become activated and start causing the disease. Yes. So using the nanoparticles, it's possible to block the viral replication. Yes. My answer will be yes.
So, okay. Vandana. So why you are not unmuting and speaking? You should unmute and ask question. So Vandana is asking, actually, I want to know that which type of drugs are impact human and animal nucleotide or the genome of both species.
Okay, so Vandana, any drug that impact human nucleotide or the animal nucleotide and both the species. So I will say, animal kingdom and plant kingdom both are very huge kingdom. And that's why we have evolution, then we have phylogenetic analysis. So anything that is very near to the human, for example, like a monkey, then maybe a rabbit, maybe mice. So generally, this is a way that any drug which comes in the market, it goes through the clinical trial. So starting that it go in the cells, then you go in the mice, then you go in the rabbit, go in the pig, go in the like a monkey, then come to the human. So any drug when come to the human, of course, it will affect a nearby animal which is close to the human. So it will be effective both in human and that is close to the human, because we don't try clinical trial directly in human. We start from the animal. I think this makes us. Thank you. So there is another question asked by Manjisha Chaudhary that what are the commonly used nanoparticles for disease based on efficacy, chitosan or gelatin or any other? Yeah, this is a very good question because gelatin and chitosan both are used in delivery system. So gelatin is very much biocompatible because mostly medicine like a capsule or tablet are in the gelatin. Chitosan has a certain extra advantage because it has like an adjuvant, like it increases the persistent type of antigen. So it has some immunological property. So when we are talking about something in terms of immunology, then chitosan has overlead on gelatin nanoparticle. So both has their different way in application using the nanoparticle and drug delivery.
Thank you very much. Now I don't think there is more question, but you can see though so many researchers have so many queries. It shows that people are very much eager to know about nano technology and its application. From question you can see how much they are eager, and that is purpose of live lecture. Otherwise, in recorded lecture, they all know about nanoparticle, but their queries, no one is there to solve and reply the queries. So that is main purpose. Introduction is required. So that at least 4 or 5 people they got their answer. So that is good. Dr. Ramendra, Pati, Pandey sir, thank you much. Dr. Saab, yes. Service to the nation. So that knowledge is percolating gradually and people are trying to learn, and so that in future they will use this knowledge, and they will also disseminate this knowledge, because I can see in online mode there are so many faculty joined, so many researchers. Manjisa madam is also faculty, this Sir Vipul sir, and so many faculties I can see here. SP Singh ji is here. SP Singh ji, आपका कुछ question है, कुछ suggestion है? Sir Prakash ji, are you listening? Okay. So finally, thank you very much. So it's अच्छा रहा 50 मिनट में आप खत्म कर दिया और 20 मिनट तक इंटरेक्शन चला, क्योंकि लेक्चर से ज्यादा इंटरेक्शन एंड क्वेश्चन आंसर इज मोर इम्पोर्टेंट as per my perception. So thank you very much. Anybody can share oral feedback if you wish, but you must write online feedback in Google form. Anything? If there is no more question, thank you very much, everybody. Good night, sir. Thank you. I will take again other, you can say, your time next time also on related topic. And anyone can interact with Ramendra Pati Pandey sir on LinkedIn. I will tag him in post. So if anybody need more help, more consultation, more collaboration, so you can connect with him. He is young, so he will help you with extra mileage. आप समझ रहे हो ना? Extra जाकर के भी आपकी मदद करेंगे. So don't hesitate to connect with him. He will help you definitely if you wish to pursue your research in this line, this topic. Thank you.
Yeah. So once again, thank you very much, Gopal ji. It was lovely interacting with all of your colleagues and students, and it was a very good session with a lot of innovative question and answer. I am always available for any further question and discussion and collaboration. Wish you all the very best and have a very good night.
Thank you, sir. Thank you, everybody. Poonam madam, आप हैं? Okay.
Yes, sir. Yes.
Final conclude कर दिए ना? कर दीजिए.
Yes, sir. Thank you so much, Pandey sir. It was really a nice talk from your side, and we are really enriched with the topic of the nanotechnology. Though I am not in that field doing any research, but I also enjoyed the topic that you have discussed with us, and no doubt we are always getting some more points from every lecture of this series. all the speakers who have been invited, and they have shared what they are mastered of. They have been discussing those topics to us. So again, in that line, today's lecture was also very much informative for us, and for that I really thanks and appreciate Pandey sir for giving or sparing his time to us and discussing these details with us, and also he tried to answer almost all the questions or all the queries from the participants who asked from him. So thank you, sir, again. And with this, we will be concluding the session for today. So with this, thank you so much, everyone. Thank you, sir. Thank you, Gopal, sir.
Thank you, madam. Thank you.
Thank you, Puna ma'am. Thank you, everyone, and have a very good night again.
Okay. Thank you, sir.
Thank you.